Hi everyone, welcome back to the channel. I'm the GCSE science teacher, and in today's video we're going to be learning about genetic engineering for GCSE biology. I hope you guys find this video helpful. If you do, feel free to give the video a thumbs up. You can share this with someone else to help them revise, and if you haven't already, please do subscribe. Thank you so much to everyone who already has. I do appreciate all of your support. So let's get started. What exactly is genetic engineering? Well, it's kind of exactly what it sounds like. Genetics to do with DNA, to do with genes, and engineering essentially meaning to make. So we're using the DNA from an organism, specifically genes from an organism, and we're transferring them and introducing them into the genome of another organism to get a desired characteristic. Genetic engineering is one of those technologies that often students find quite tricky to understand. So this is the definition you need to know. Essentially, it's changing the genome of an organism by introducing a gene from another organism, resulting in a desired characteristic being expressed phenotypically. Those words I've put in bold for you are keywords in this topic that you need to understand and know. If you get stuck with any of these, let's just break it down. The word genome is talking about all the genes in one specific organism. So the human genome, for example, is all the genes that make up a human, and other organisms, plants, animals, bacteria, will have their own specific genome. Organism is just a living thing. A gene is a section of DNA found on the chromosome. If it's a eukaryotic cell, it doesn't always have to be on a chromosome, because remember, bacteria have genes as well. Desired characteristics. So this could be anything. And usually with genetic engineering, we're talking about plants. There's loads of examples where plants have been genetically modified or genetically engineered to have disease resistance or have really large fruit yield, or in the case of golden rice, this was an incentive that was essentially taken out where beta carotene was introduced into rice. It gave it a kind of a yellowy golden color, and this increased the amount of vitamin A that was available in those particular plants for people who in different parts of the world didn't have access to different kind of fruits and vegetables that had a lot of vitamin A, and they were seeing a lot of deficiency issues there, so things to do with like blindness. So this was a really good way of tackling that issue. But obviously that's not the only thing. Then we'll talk more about the risks and the ethics around it too. And then phenotypically as well. Phenotypically is just talking about the phenotype, so the physical features based on the genetics that we've just discussed. So as I mentioned there, I said about genetic modification, genetic manipulation, GMO, GM. You may have heard of one of those versions. This essentially is a way of discussing the same thing. So that's what we mean by genetic engineering. And we're going to discuss this in a bit more detail now. So how does this actually happen? And this is the process that you need to know. So it may be worth writing a flashcard of this. You might want to pause the screen and make a few notes as we go through. So the first thing that happens is that scientists will select a desired characteristic and the gene is isolated. Isolated just means that it is identified and it's the one that scientists are going to be interested in taking. So an enzyme is used to essentially cut out the gene that is responsible for the characteristic on that chromosome, the one that we're interested in. That's how we isolate it. That's how we take it out. We use enzymes to do this. This gene is then transferred and inserted into what we call a vector. Now, you've heard this word before in the infection response topic, where we learn about vectors with malaria, for example, which is just an insect that carries a protist disease or a disease in general, or a pathogen causing a disease, I should say. But in this case, a vector essentially is just something which will carry something else. So that's what we mean by vector in this scenario. So that could be bacterial plasmids, it could be viruses. And it's at this stage, I would mention it would be worth looking back at the structure of bacteria. So when I use the word plasmid, I'm talking about that extra loop of DNA that bacteria have. And often they will have genes on those plasmids that help with antibiotic resistance and things like this. But that's more of a recap. So then the next step, the vector inserts the gene into cells during early development, and the modified organism will replicate this particular gene over and over and over again until the gene that has been inserted is expressed and the desired characteristic develops. And you can see that lots of copies of that gene will be made. So some of the benefits, of course, if we introduce a desired characteristic, we might have an increased crop yield, insect-resistant crops, disease resistance, insulin production to treat diabetes, as I said before. It's much faster than selective breeding, because obviously selective breeding requires generational change. Also, it could improve food security, and it could reduce diseases if we could genetically modify different vectors like mosquitoes, which I mentioned, and this could reduce the transmission of malaria, for example. And it could help with deficiency diseases as well, which we already see in the case of golden rice with beta carotene. So there's lots of different benefits, but the risks are ones to consider as well. So some of the risks include selective genes may damage the host organism. We wouldn't know until we do this. Some people may feel it's unethical to change the genome of a species. It can be very expensive to develop GM products in some countries, and it can be potentially harmful, so toxins from the crops. It could also lead to allergic reactions. Now, how does this relate to the GCSE? What could they ask you to do? Well, they could ask you to evaluate genetic engineering. They could give you some data about the positives and negatives. They could ask you to just discuss the positives and negatives through an evaluate question. And it's for you as a individual to come up with your own conclusion. So remember, when you are doing an evaluate question, you want to give the positives, the advantages of this process and the disadvantages, but then also come up with your own conclusion at the end. And there really is no right or wrong answer here. You can have your own opinion and you will not be penalized for being pro or anti genetic engineering, as long as you can debate your point, because that's one of the skills they're looking to assess you on. They also could ask you in this topic what genetic engineering is and give some examples. You always want to go with things like diabetes treatment and insulin resistance. You always want to go with things like crop yield or disease resistance in crops. Beta carotene has often come up as a topic in the exams as well. So my biggest advice for this topic is to know the process, know the keywords, because that's what they're going to look for in the mark scheme, and also be aware you could be asked to write a 6 mark to 4 to 6 mark answer, which is an evaluate question usually in this case. I hope you guys found that helpful. Thank you so much for your time. I've been the GCSE science teacher and you have been curious. If you did enjoy this, let me know by giving it a thumbs up. Feel free to stick around and join this amazing community by subscribing. Share this with your friends, share this with your family, anyone you know that could benefit from learning more about science and GCSE science specifically. It would be great to have you guys here. So thank you so much in advance. And if you have any video suggestions, I know we're in the new year. So if you are looking to revise, maybe you're in year 10 or even year 11 and you're doing the real exams this year, let me know. Best of luck with all that. And I'm more than happy to make very specific content if you'd like support with how to revise science, if you would like support with 6 mark questions, if you would like support with science and maths questions like graphs, variables, all of that. I do have lots of videos on my channel, but if you have a look through and you think to yourself, oh, actually, I would really benefit from a specific video about this, do let me know in the comment section. It's always great to hear from you guys. And in the meantime, have a fab day and I'll catch you in the next one. Take care. Bye.